And the next speaker is Dr. Einar Sigurdsson from New York University, and he's talking about the clearance of pathological tau protein with the immunotherapy.
Yeah, let me first of all thank the organizers for the invitation, Aubrey and the foundation. It's a pleasure to be here. And I would like to start off by acknowledging the people in the lab that did most of this work. The initial experiments that I will talk about were done mainly by Asuni when he was a postdoc in the lab, and subsequently subsequent studies by Boutajangout and Pavan Krishnamurthy, with great help from Hamitha, Raya Mohammed Said, and Johanna Ingadóttir. So yeah, I just wanted to disclose that I'm an inventor on NYU patents and applications related to this technology. And so with aging, there's accumulation of a variety of amyloid proteins that are likely to be toxic. And I just wanted to highlight here briefly those that are currently being investigated as targets for immunotherapy. And today I will mostly speak about the tau protein, but I've also been working on the amyloid-β and the prion protein and the islet amyloid polypeptide in type 2 diabetes. Now, there has been sort of renewed interest in tau as a target in Alzheimer's disease because of rather sort of disappointing outcome of the amyloid-β immunotherapy trials in Alzheimer's disease. So, for those of you that are not too familiar with the disease, there are basically two main proteins or aggregates, the amyloid-β and the tau protein. And in the initial A-beta immunotherapy trial that was designed to clear these A-beta aggregates, they noticed that when these individuals came to autopsy, they noticed that there was a very extensive clearance of amyloid-β from the brain. They had a high antibody titer against A-beta, but they were completely demented. So it did not seem to affect the progression of dementia, at least not in these a few individuals. Now, in all fairness, this trial was halted, so it's conceivable that there was a decrease in the antibody levels over time, and it was just not sufficient to stabilize or affect the progression of dementia. So there are several ongoing trials targeting amyloid-β, both passively and actively, and we should probably know sometime next year if this approach has any efficacy. But one of the main reasons why it may not be effective is that it may be too late to target amyloid-β at this stage of the disease. So if you just look at sort of briefly what may be happening in the disease is that under normal conditions you have soluble A-beta that is being released at the nerve terminals, and it's likely to be a neuromodulator and may also have some neurotrophic effects. With aging, and particularly in Alzheimer's disease, there is accumulation of A-beta oligomers, likely because less of it is being cleared for a variety of reasons. And as these oligomers, protofibrils, start to accumulate, they damage the synapses. They cause an activation also of microglia and astrocytes, and there's an increased calcium influx into the cells, activation of protein kinases, hyperphosphorylation of the tau protein, and eventual tangle formation in the cells. So the tangles are basically the end stages of the tau pathology. And when you're targeting individuals that already have cognitive impairments, you're most likely targeting at this stage here, where the damage may be too far gone when you target A-beta, that there are secondary pathways that have already been initiated, and it may be too late to do much about it when you're clearing A-beta at this stage. However, A-beta may be an ideal target as a prophylactic therapy. So if you look at likely scenario, we know that A-beta is very important, at least in the cases where you have a mutation that increase the generation of A-beta. So if you assume that A-beta deposition starts first in the disease, we know that it likely plateaus out based on both autopsy studies that do not see a good correlation between the degree of senile plaques and the degree of dementia, and also based on new imaging studies that where they have been able to follow these patients with PET scans, and they see that there's a progression of dementia, although the signal in the PET scans is constant. Now, tau pathology likely correlates much better with the degree of dementia based on autopsy studies that it most likely because it may be a later phenomenon and is more directly related to the sort of the loss of the cognitive functions. So it may be better to target tau pathology at this stage in order to be able to affect the cognitive impairments, whereas targeting A-beta may be more appropriate at the earlier stages. So the tau protein, just briefly, it's the problem with tau is that it's a much larger protein than A-beta, so it's conceivably more difficult to target, and also it's mainly intracellular, and that's also causes difficulties for drug targeting. So it's expressed primarily in the brain, and it binds to and stabilizes and promotes the assembly of microtubules. So it's important for axonal transport and for cytoskeletal integrity. And phosphorylation of this protein reduces its binding to the microtubules, and it causes it to be more hydrophobic, and it starts to aggregate. So it's analogous to A-beta aggregation, that these aggregates like to form oligomers early on, then protofibrils, and eventually paired filaments and tangles. So our approach has been to target these phosphorylated entities with immunotherapy in order to try to clear them so that we do not have the subsequent toxic effects. Now, initial studies, we did active immunization, which is where you immunize with an immunogen and you induce the body to make antibodies against the immunogen. We used immunogen targeting this region. Was a very immunogenic region of the molecule, and there are already antibodies available out there that are used to diagnose the disease that have a high affinity for this region here. So we immunized animals that had a tau mutation, mainly because it's a more aggressive form of the disease, and at this time, this was in the early 2000s when we started these studies, there were not very many models available. This was the first model that was developed by J. Lewis and Mike Hutton at the Mayo Clinic at the time. Now, we got a good antibody response, and we also noticed improvements on motor impairment tasks. So these animals developed tau lesions extensively in the brainstem and in the spinal cord, but also in cortical areas and hippocampus. And with associated with the lesions in the spinal cord and brainstem, they have motor impairment, develop paralysis over time. So it's a very good functional outcome measure to look at their performance on a variety of motor tasks. And we noticed that immunized animals, shown here in red, they did much better on a rotor rod task where they have to be able to stay on a rotating rod, that the speed increases over time. And both at 5 months and when we repeated this at 8 months, they were improved over the control animals. We noticed that there was a decrease in insoluble tau, not quite significant, but there was a dramatic increase in soluble tau, suggesting that it was being mobilized from its insoluble to its soluble form. And there was a substantial, dramatic decrease in pathological tau in the brain in a variety of brain regions. And I'm just showing you some examples here from the paper. So this was all very positive, but it was unclear what the mechanism was. So what we initially did is that we took antibodies from basically this high titer mouse. So we isolated the antibodies, we labeled them with a fluorescent tag, FITC. We then injected it into the carotid artery of P3ONL mice to see where these antibodies would end up. And they easily got into the brain, and we noticed 1 hour later when we sectioned these, the brains from these animals and co-stained them with DAPI to detect the nuclei, we noticed a pattern that it appeared that these antibodies, they definitely got into the brain, and they appear to be within the neurons. So when we took these sections, showing you some examples here, and we then put tau antibodies onto the sections, we noticed that the injected antibodies, they co-localized with tau, suggesting that they were being taken up into cells and binding to pathological tau in the cells. Now, we did a variety of control studies. We used also just normal IgG that we labeled, injected into the same kind of mouse, and we certainly we saw it get into the brain, but it was a non-specific binding. It was not inside the neurons. And the FITC, phospho-tau, the anti-tau antibodies in wild-type mice did not get into the brain in any appreciable quantities, and the same with the control. And that's because they have an intact blood-brain barrier. It's very limited. You only get about 0.1% of antibodies into the brain under normal conditions. But these tau pathology mice, they had presumably inflammation in the brain that led to opening of the blood-brain barrier. And that's important also for therapeutic implications, that antibodies probably get into the brain in sufficient quantities to treat neurodegenerative diseases. And this has been sort of well established in the A-beta trials, that they definitely see clearance of the plaques in the brain of these individuals. Now, we wanted to follow up on this in a slice culture study so that we could use a little bit less of these antibodies and try a different sort of preparations. So then you take the brains basically from a mouse with the mutation that already has the pathology, and you slice it up and you incubate it with oxygen, and you then add the antibodies directly to these slices. And then you section them into 40 microns. So you have about 400 micron slices and section them then into 40 micron sections. So what we noticed is that these antibodies were definitely getting into the brain, analogous to what we had seen in the in vivo study. And then when we took markers, we took these smaller sections that we then added antibodies to. We saw that they were co-localizing with markers, lysosomal and endosomal markers, and also again with antibodies against tau, different antibodies here that recognize pathological tau, that there was a co-localization of these injected antibodies, suggesting that they were getting into the cells, they were binding to pathological tau, and that this, presumably the clearance mechanism was happening via the endosomal lysosomal system, and very limited staining in wild-type animals. Now you can see here this high-power magnification of a confocal image is that the antibodies are getting into what appear to be neurons, and they're co-localizing with a lysosomal marker LAMP2 in the neurons. So we wanted to follow up and confirm this by biochemical analysis. So what we did then is that we took, we basically made lysosomal fractions from the brain. This shows you different animals here, different fractions that have lysosomes in them as recognized by LAMP2. And we then had antibody that is supposed to be specific for FITC IgG, which is then the material that we had added to the slice culture, so basically the same conditions as in the immunohistochemical study. And we noticed these antibodies in the lysosomes, and then when we reprobed with tau, these blots, we noticed that these blots also had plenty of tau. So this is basically giving us the same results as in the immunohistochemical study, that these antibodies are getting into the cells. They're getting, we find them in lysosomes, and tau are also in the same location. And we also just looked at these animals. These are the P3ONL mice. This is the name that was given to these animals. And they have more lysosomes than wild-type animals. We can see a dramatic increase in lysosomes in these animals, suggesting that they are maybe upregulating the lysosomes in order to try to get rid of these aggregates. Now, the mechanism, it's not entirely clear, but there's likely that there are several mechanisms at play here. We know that there are several receptors for IgG on all cells, including neurons. This is part of your immune system, that you need to have, antibodies need to have access into cells and to pathological conditions to clear pathogens. And normally this is not in use, so in a way what we're doing is that we're sort of hijacking the system. And when we have antibodies, so any antibody would be taken up, but most of them would be released back into the outside. But in our case here, that this is just a very primitive sort of explanation of this, that tau is presumably, tau aggregates would be contained within autophagosomes. This has been shown in P3-1L mice, that there's an increase in sort of lysosome or autophagosome-like structures in the mice that have these aggregates. And the antibodies would be taken up into endosomes. These would then fuse with autophagosomes, and the antibodies would bind to the tau aggregates, disassemble the aggregates, and thereby facilitate access of lysosomal enzymes to degrade these aggregates. Now, it could also be the antibodies might actually get into basically sort of directly through damaged membranes, bind to tau, and this complex would then be engulfed into autophagosomes, and the same process might happen. Now, there was a renewed interest in this. So our data basically supports the intracellular pathway, but we can't exclude the extracellular clearance. We would need a different model system to look at that. And we're currently looking into this in collaboration with Mark Diamond, who was one of the first ones to, basically the first one to show that extracellular tau is likely to be important for the spreading of tau pathology from cell to cell, in a way sort of like a prion-like mechanism that may also apply to all of these amyloid neurodegenerative diseases, is that it's likely to, that these cells are trying to get rid of these aggregates, spitting them out, and then they're taken up by adjacent cells. And this made this much more attractive for the pharmaceutical industry, that because they realized that, well, we don't necessarily have to get the antibodies into the cells that can conceivably cause some problems. We may just be able to block the extracellular transmission. And so this would be analogous to targeting amyloid-β, which is mainly extracellular. And we already know that that works, at least clearing that works in AD, even though it may not have beneficial effects when you start late. Now, there is another mechanism that has not really been explored, but there was a recent study pointing out that there's a high affinity antibody binding site on a ubiquitin lipase called TRIM21. And this may be important for clearance of viral particles. And conceivably, this may also be involved in facilitating clearance of misfolded tau proteins. The ubiquitination of tau, misfolded tau, is known to take place and is then cleared through the protosomal degradation. The protosome is too large to clear these larger aggregates, but conceivably antibodies might facilitate this clearance, although that's something that remains to be investigated. So several different pathways at play. Now, we wanted to see if tau can prevent cognitive decline. The other model that we had been using, it's very good to look at, to use a motor impairment as an outcome measure, because it's quick and doesn't take a long time to do those tests. But cognitive testing is a little bit more elaborate, but we wanted to determine if we could also see cognitive improvements in a different model. So we focused first on the AIDS tau mice developed by Karen Duff and Peter Davis, because their pathology, the distribution resembles Alzheimer's disease better than in the JNPL3 mice. Now, we actually ended up using a different model. We crossed these animals with a PS1 mutation mice that we noticed that they had accelerated tau pathology, so it would sort of make these studies somewhat shorter to do, and we decided to move ahead with those studies in this particular model here with the accelerated tau pathology. And we did a variety of, so we treated them in the same way with the active immunization for several months, from 2 to 8 months of age. And then we looked at cognitive tests. The radial arm maze, we noticed that they were dramatically improved, had much fewer errors than all the control groups. And the control groups being identical animals. And then also litter mates that have mouse tau that actually interferes with the pathology. And the AIDS tau mice that have less pathology than the ones that have the PS1 mutation as well. So the immunized animals did much better. They also did much better on an object recognition test. Animals have to, basically wild-type animals spend about 70% of the time with a novel object, and we noticed the same thing in the immunized AIDS tau PS1 mice, whereas the identical controls spend about 50%, suggesting that they were not really recognizing the novel object or distinguishing it from the old one, remembering that it was an old object. Closed field symmetrical maze is the third test that we used, and the same results, that they have much fewer errors in all these tests. So the animals basically have to find their way from one side to the other side, and if they go into this part here, that's scored as an error. Then there are multiple trials, they go back and forth. And so it's very clear that this is beneficial in this particular model, at least. And we looked then at the pathology in the brain, and we saw a dramatic decrease also in tau pathology, focusing on the piriform cortex mainly because it's very easy to analyze large neurons. And we saw a dramatic decrease there. We saw the same pattern in Western blots that I don't have time to go into, but this has been published recently in Neuroscience. Now we then wanted to see, so we have been using this immunogen, 30 amino acids, phosphorylated at 396 and 404. We wanted to see if we could get the same effect by using antibodies against the same region. So Peter Davies had generated the PHF1 antibody many years ago, and he was generous enough to collaborate with us and give us large quantities of this antibody so that we could immunize animals with it. And I will just cut this short. I will not go into the details of the immunization process, but we published this recently in Journal of Neurochemistry. So we noticed the same kind of results, that we're getting a decrease, basically functional improvements, that they were doing better on functional tests. So this is back to the P301L model. There was a decrease in pathological tau by immunohistochemistry and also on Western blots using 2 different antibodies. The antibodies, they go down over time. That's sort of typical, that you know, the half-life is few weeks in these animals. So you need to give multiple injections. And we saw a decrease in the pathology, and we noticed that the animals that had the highest antibody levels had the least amount of tau pathology, suggesting that you may be able to tailor this somewhat in individuals that increase the dose depending on what the plasma levels show. We're giving this also intraperitoneally, and you might be able to get a more consistent plasma levels by giving this IV. Now, so this basically supports, to summarize, you know, what we have at this point is that we're definitely seeing a reduction in the accumulation of tau aggregates in 2 different tangle models. It prevents and reverses cognitive decline in this particular model here, and attenuates motor impairments in this model here. We have some data on the reversal that I did not have time to go into. Both active and passive are effective, and we also have data showing that you can target more than one epitope, and I don't have time to go into that. So we're making antibodies because we want to test a variety of different antibodies. It's probably a little bit safer to use the antibody approach in initial clinical trials, because the active, there might be some T-cell side effects that you would like to avoid. But I think sort of chronically in the future that the active approach is probably the way to go. But as a proof of concept, the passive is more attractive. So we have been making antibodies. There's some epitopes. These are the same epitope as the PHF1, and we see that they're phosphor-specific. They're recognized in the 404 region. This is they're not the 396. We also have non-phosphor-specific, and it's important to compare these antibodies to see if they have a similar effect, or if there might be some toxic side effects associated with this one, because this one would recognize normal tau also. And then we have antibodies against, for instance, the 262 that are highly phosphor-specific. And they recognize pathological tau predominantly on Western blots, and a very limited reaction with wild-type tau. So we are doing studies, in vivo studies, with these antibodies, showing you here the phosphor-specific analogous to the PHF1, And we're doing now, trying to do a reversal study where we start the treatment at 8 months when we know, based on the previous study, that we already have extensive cognitive impairments. And we see that in these animals that they do much better on radial arm maze than the controls. They do much better on the closed field symmetrical maze on all 3 different tests. And they also do better on object recognition. They recognize the novel object. So this is analogous to what I showed you with the active approach with a novel antibody that we are developing. And we have antibodies now against several different epitopes that we are currently characterizing and preparing for studies in other animals. And so just to summarize, I think that tau immunotherapy is a very promising approach for not just Alzheimer's, but other tauopathies as well. And now several groups have confirmed our findings. Most recently, Rosenmann's group published in Experimental Neurology in 2010 using active phosphoimmunogens showing similar results. And Martin Citron at Eli Lilly just published a couple of weeks ago in JPC using some of Peter Davis's antibodies, basically seeing the same effect that we were seeing. And several other groups have been reporting at recent meetings similar results. And I think that this is particularly attractive in Alzheimer's disease because the tau pathology correlates much better with the degree of dementia, suggesting that it may be more efficacious at this stage of cognitive impairments to target tau than to target A-beta. And finally, I would just like to acknowledge again everyone in the lab and colleagues at NYU that have contributed to the studies, and I would like to thank Peter for his antibodies, and Peter and Karen, Jada Lewis and Mike Hutton for making their animal models that we have been using. And support from the NIH, the Alzheimer's Association, the ATDF, AFTD, and the Hearst Trust. Thank you very much.
This paper is now open for questions. We have time for it.
Hi. So in all your studies, you are targeting the tangle protein. And as you're certainly aware, there's a bunch of studies, both in animal models and now some cell models to help explain it, suggesting that even the wild-type protein can act as an effector of the neurotoxic effects of A-beta. So when A-beta is present, it causes missorting of tau across the neuron, and that seems to be causing a lot of the dendritic pathology and so forth. So do you have any thoughts about the relative importance of clearing out the tangles versus earlier on doing something to block that toxic effect of that is mediated by the wild-type tau from A-beta?
Well, I'm not necessarily sure that it has been established that wild-type tau is mediating it. But when you knock out tau in A-beta models, you have less pathology. And it is conceivable that there might be, I think that A-beta is clearly causing some tau pathology, and that's, as you mentioned in cell studies and in animal models. And it's conceivable that it may be, you definitely see some hyperphosphorylation of tau, wild-type tau in the A-beta model. You see it in the dystrophic neurites. So it's in a way sort of analogous, but you don't see the late events. You don't see the tangles. But I think that what we are doing, you can target the oligomers, you can target the smaller aggregates. The larger aggregates are probably more difficult to clear, and we don't see a complete clearance in our models. We see partial clearance, and that's probably because we're primarily targeting the smaller aggregates that are easier to clear. So I think that the therapy can also work on, well, wild-type tau is pathological in AD, that there are no mutations of tau, so you are in a way targeting that as well. I don't know if that answers your question. Yeah.
How will you avoid recapitulating the mistakes of the A-beta immunotherapy, since mice are deficient in complement receptors that humans have? And in the human studies, there was a massive meningoencephalitis induced when with the antibody therapy.
Well, there was no encephalitis with the antibody therapy, but there was with the active immunization, that is when you immunize them with A-beta itself. And so when you use a large molecule that has, a T-cell epitopes on it, you can get side effects related to that. I'm not sure if it is necessarily related to complements, but it's certainly true that the complement system is very different in rodents and humans, and that may have some implications. But the antibody therapy so far is showing very limited side effects. With high doses of A-beta antibodies, you do see what may be microbleeds in the brain, and that may be related to the amyloid angiopathy, although that's not entirely clear. Now, with tau antibodies, they would not be targeting amyloid angiopathy. There are some dystrophic tau that is associated with blood vessels that may conceivably be affected, but that's unclear at this point if that would have any sort of particular relevance or side effects associated with it. And I think that the antibody therapy is probably safer as a proof of concept because you avoid the T-cell side effects that are a little bit, that are more difficult to control.
I think it's important to characterize the nature of the tau models. I'm concerned that they don't model the age-related component of Alzheimer's, and I think that's the case in some of the amyloid-beta models as well. So if you could just talk a little bit about that and explain exactly how they model the tau. Is that overexpression? Is it as simple as that? I guess as a strategy to focus on tau when it's more associated with dementia but comes towards the end of the course of the disease, it seems almost counterintuitive. Presumably you'd want to try and nip it in the bud earlier on, and even later when you start seeing tau pathology, you'd want to try and get back to the causal origin of the disease to affect the tau and the amyloid. So those are my questions.
The models, the initial model with the tau mutations were basically to model frontotemporal dementia. And they noticed these tangles and that you also see in Alzheimer's. So there's sort of an overlap there. The pathology was, in the initial models, was in regions that led to motor impairments, primarily in the spinal cord and brainstem. You do see cell loss in these areas that sort of fit with what you would see in these diseases. The other model that I showed you, the AIDS tau model, when it was initially published, there was very limited pathology because the mice were expressing all 6 isoforms of human tau, but they were also expressing mouse tau. So they then noticed that when they crossed these animals with animals that didn't have any mouse tau, they saw the pathology. So it seemed like a mouse tau was interfering with the development of pathology. Now there are some other models that do not fit into this because they actually do see pathology with mouse tau. And mouse tau in those models may be actually seeding the pathology. I think Mandelkoff's group has some data on that. So it's quite complicated. But that particular model, the AIDS tau model, Peter Davis recently published that they do have cognitive impairments and there appear to be some cell loss in them. We are looking at the AIDS tau PS1 mutation model that we developed in the lab by crossing these two. We don't know yet if there is extensive cell loss, but we definitely see cognitive impairments in the animals. And so it's a partial model of AD because we do not have A-beta aggregation in them, but we're also looking at that by crossing them with mice that have that. So it's kind of unclear at this stage how well they sort of recapitulate the disease, but I would say it's a good model, to test these therapies, initially.
Okay, thank you.
Okay, very good.
The next